So next, we will discuss about transgenic mouse model and gene editing. So you probably heard a lot about transgenics, transgenic mouse model, or knock-in, knock-out mouse models. And this has been very important for investigating mechanisms of human disease in a mouse model. And why we study this in mouse? Because mouse is easy to manipulate for this genetic aspect compared to human patients. So like in vivo experiments, many are done in mouse or rats or monkey, like these non-human animals and vertebrate animals. So I want to explain you two kinds of this model using genetic modification. The first is transgenic mouse, and the second is knock-in or knock-out mouse. So they are a little different. So transgenic mouse is a recombinant DNA is inserted randomly. So let me see here. So this is an egg, a fertilized egg of a mouse, and we inject the gene into pronucleus of a fertilized egg, and that zygote, which is a fertilized egg, into host mouse, and that will create a transgenic mouse. And we can use this as a disease model or interested in specific genes expression. So this recombinant DNA is inserted randomly into the genome of the oocyte. So the transgene will be expressed. However, there's a little bit of a problem, which is transgene expression may occur in an uncontrolled fashion because we do not know where this recombinant DNA will be inserted into the genome, which has been random. So we have developed another way of knock-in or knock-out mouse, which is we put recombinant DNA in or specific genes, we take it out. So that's coming from stem cells from embryo. We take the stem cell and transfer genes into the embryonic stem cells in vitro and select those stem cells into the blastocyst of a mouse and those into the host mouse. And the host mouse will create a mixture of these. So the chimeric mouse is born and you self-breed and breed. Then you can see the progenies are genetically altered. You can find out into which ones have the pure knock-in or knock-out mouse. In this case, we define the genetic modification in a target vector with a drug resistance selection marker so that we can select them and homologous regions of a genome to define specific region of integrating into the genome. So gene expression is now under the same control as for the endogenous wild type layer. So knock-in or knock-out mouse is a good model for mimicking a human disease. Now, recently, more interesting specific gene editing tool has been discovered, and this is called CRISPR-Cas9. You probably have heard many times. And this CRISPR stands for clustered, regularly interspaced, short palindromic repeats. And palindromic, I put as an example, such as in one side reading and the other side read the same. So GAA-TTC from this 5 prime. On the other side, GAA-TTC is the same. And Cas9 enzyme is a CRISPR-associated nucleases. And nuclease is an enzyme that cleaves the chains of nucleotide in nucleic acid in small units. So how this CRISPR system, originally derived again from bacteria, works to correct potentially genetic defects in humans. So an RNA guide molecule can be programmed to match any unique DNA sequence in human genome. So that is carried by a special enzyme Cas9, which you can attach to the RNA guide, and it will find out the target sequence of DNA. And when it aligns with the target DNA sequence, this Cas9 enzyme will cut the both strands of the DNA double helix, and then this can be amended by extra DNA of our interest to insert or delete defective DNA. So this is a precise gene editing tool which potentially revolutionized our biomedical research. I also want to introduce one nice, interesting movie related to this, a film which has been opened in 1997 and called Gattaca. And this film is a future, describing future society by eugenics, where potential children are conceived through genetic manipulation to ensure they possess the best hereditary traits of their parents. So what if in the future, if we can manipulate the genes of our progeny, maybe very rich people can create those clone humans. So when they have problem with their liver, their heart, they may want to replace from these clone humans. So this concerns over reproductive technology, and which of course is we don't know in the future whether this will happen, but this gives us a good idea how genetic engineering can influence our human race. And you can see this title, Gattaca, you now recognize. This is composed of ATGC, which is a nucleotide. So we have studied this time the central dogma of molecular biology and transcription and translation and gene cloning and recombinant DNA technology. And I hope you can understand more of this by reading papers and your own studies.